Good evening, everybody. Hope that you guys are doing all right. I have one announcement that I need to make, and that is related to next week's classes. I have some conflicts on both Monday and Wednesday, and so for next Monday and Wednesday's classes, um, I'm doing these. I'm using this piece of software called Edpuzzle. Um, and so I'm pre-recording the lectures, but then I'm using Edpuzzle to basically integrate questions along the way throughout those. So I'm going to make those available to you hopefully before the end of this week. Um, Now, with that in mind, if you want to meet during office hours or some other time next week, I can do that. I just have these kind of unavoidable conflicts next week, and I apologize for that. Um, so that's kind of one difference for next week. And I'll send out an announcement on Sunday detailing like, hey, here's these things that are going to be posted and when they're going to be due. Now, with that in mind, next week is our week before spring break. Um, so we will have that, you know, week before spring break and then actual spring break. Um, the assignments that are going to be assigned next week will not be due until the Sunday before, or sorry, the last Sunday of spring break, which is March 16th. Again, I'll detail all this on Sunday in an announcement. Um, but yeah, I just wanted you guys to know that. Um, and there won't be anything that's actually going to be assigned to be submitted during your spring break week. So that's one thing that I wanted to clear up. So like I said, on Sunday I'll post an announcement with, hey, here's what's going on. Um, and like I said, if you want to meet at any point to get caught up or anything like that, or if there's something that you want clarification on, I'm glad to do that. But for next week's classes, those are going to be asynchronous classes. Do you have to do them during that scheduled time? No, you don't. Um, you can do them, they'll be up as long as the internet's up. So thank you, and we'll proceed from there. Now, with that, got my first poll question for you. Glucose is an aldose, a ketose, a pentose, an allose, and oh, not that one. There we go. I'll be sharing my screen. Share. Okay, we got sugars again, and we got 51 folks reporting in, and they are saying that it is an aldose. I'm gonna go ahead and stop right there. Um, we had 43 people say that glucose is an aldose. You are correct. Glucose is a number of different things. In addition to being an aldose, glucose is also a hexose. Um, and sorry, my monitors are doing a weird thing where they've switched. Um, so glucose, maybe one of the most important sugars that I want you guys to be familiar with. We have the aldehyde functional group up top. That's carbon number one, two, three, four, five, and six, CH2OH. Now, whenever we look at a sugar, so this is kind of my general backbone for a aldose. And then my next question that I want to share with you guys, and I got a message. Um, okay, go ahead and make sure that you answer this one then. On a linear pentose, which carbon will identify or which carbon will reveal if this sugar is a D or an L sugar? And I'm going to go ahead and draw backbone. So we are looking at 46 folks have responded. You can give it another couple ticks. Okay. So 55. Assume that. Okay, we're going to go ahead and end it right there. Share the results. The two most popular answers were 4 and 5. The correct answer is 4. So whenever we look at a pentose, what we're looking for is the highest chiral carbon. So pentose has 5 carbons total, and our highest chiral carbon is carbon number 4. Okay. So right now, what I've got a little, I didn't do that. I apologize. Not that. There we go. What we've got is our backbone for glucose as well as I'm going to go with fructose. Now, we'll start out with glucose. This is carbon number 1. This is carbon number 6. I'm going to draw these linear, and then I'm going to translate that into the cyclic form. So if we look at all of these different carbons, we're not too interested in really any of them. I mean, it's helpful to be able to see, okay, well, carbon number 1, and just like the other day, I'm sorry to interrupt myself. That's okay, Joel, don't worry. Um, if you have any sort of issues, I am presenting a PowerPoint right now, so if you don't see that, please send me a private message and I will go ahead and unshare and then reshare again. That seemed to resolve the issue the other day. Um, Now, when we look at glucose, we've got an aldose. This is an aldohexose at this point in time. So this backbone right here on the left-hand side, I could call this an aldohexose. That information is all that I have right now. Now, whenever I throw this OH here, now I can call this a D-aldohexose. I can call this a D-aldohexose because this hydroxide group is on the right-hand side of my molecule. Now, I'm going to fill in the rest of these and indicate that this is glucose because on carbon number 2, 3, and 4, I have hydroxide groups on the right-hand side, left-hand side, and right-hand side. I've got hydrogen, hydrogen, hydrogen. Okay, so there is glucose right there. Now, one of the things that fructose is commonly referred to as is it's referred to as the ketose version of glucose. And so if we look at glucose, we've got our functional group right up top here. If we look at fructose, here's our functional group, our ketone functional group. Now, with that, this structure right here, what I can say about it, it is a ketohexose. Is it a D or L ketohexose? At this point, I can't say. However, as soon as I go to my second highest numbered chiral carbon, then I can go ahead and designate it as a D or an L ketose. Now, with that in mind, it's important to be able to identify, oh, is this a chiral carbon or not? When we look at this right here, why is it doing that? Okay. This, and this are all chiral carbons. Carbon, this one right here, OH. Now, this is a D ketohexose. And just to kind of replicate what we have for glucose, we've got OH, H, OH, H. So that's drawing fructose, and that's basically drawing our ketose version of fructose. Now, the next thing that I want to do is I want to go ahead and translate my cyclic or my linear form of glucose to the cyclic Haworth projection. The Haworth projection of glucose, and this is gluco pyranose. I mentioned that cyclic sugars can either be a furanose or a pyranose. A pyranose is a cyclic sugar with a six-membered ring. Now, one of those members, the way that I always draw a Hayworth projection, what I always do is I make my oxygen, looking at it in terms of like the face of a clock. I kind of put that in like a, I don't know, like a 2 o'clock position, 1 o'clock position, basically, a little bit over. And then I draw my carbon 6 CH2OH. Now, what I have here is my six-membered ring. One of those members is oxygen. This right here is my new chiral center. That hydroxide group is either going to be pointing up or it's going to be pointing down. If that's pointing up or it's pointing down, it doesn't influence whether or not this sugar is glucose or something else. Instead, what it influences, are we talking about alpha glucopyranose or beta glucopyranose? Somebody messaged me a moment ago and said I am unable to see the PowerPoint, but they didn't give me an update if they could now. So if you don't mind checking in. Um, so then this is carbon number one. I'm going to erase my arrows, swinging that up. This is carbon number one. This is my anomeric carbon, my new chiral center. This is carbon number two. This is three. This is four. If I look at my linear form of D-glucose, arguably it's carbons 2, 3, and 4 that I'm most interested in. And I have carbon number 2 on the right-hand side of my molecule, carbon number 3 on the left-hand side, or my hydroxide group on the left-hand side of my molecule, and 4 on the right-hand side of my molecule. Okay. So that translates to, that's okay, that's all that I'm showing right now. This is a part of a PowerPoint. Um, we've got this going down, up, and down. So right, left, right translates to down, up, down. Okay. Being able to take a linear molecule like glucose and translate it to the cyclic form is very helpful and very powerful. Glucose typically and sugars are typically going to exist in their cyclic forms. Now, it's also very important to be able to identify a D sugar versus an L sugar. The question that I asked a little bit ago about a pentose and the highest numbered chiral carbon. Now, in this case, we're looking at a hexose. The highest numbered chiral carbon is carbon number 5. But when you shorten the sugar, it's only 5 carbons. Now your highest numbered chiral carbon is 4. If we're talking about glyceraldehyde, a triose, then our highest numbered chiral carbon is, which is also our lowest numbered chiral carbon, but here we are. Okay, so these were just some high points that I want you guys to have like a little bit of a refresher on and that you feel confident on. And I believe in you. And so we're doing fine. Now, with that in mind, today's learning objectives, we're going to look at being able to identify a disaccharide when given the two sugars and know the type of linkage. List the biological roles of glycogen, starch, and cellulose. I want you to be able to name the sugars and linkages that make up glycogen, starch, cellulose, and chitin, and name the cellular locations where protein glycosylation and glycoprotein transport occurs. Name the amino acids that are commonly used to link to carbohydrates, and describe the roles of glycosaminoglycan polysaccharides in organisms and cellular processes, specifically ground substance and blood clotting. Now, this is one of the challenging things to me about a biochemistry class is that you go so deep sometimes on a molecule that you're like, oh crap. Am I expected to be going so deep on absolutely everything? What the heck? Blood clotting? What's in blood? Um, and to answer that question, we kind of, in this class, we kind of go super deep and then we pull back. And then we kind of exist at the surface, and then we go back, and then we exist at the surface. So, in order to address our first learning objective, I want to go back with the last thing that we talked about, determining if a sugar is a reducing sugar. This is something that's a little bit challenging to kind of wrap your mind around, but what I want you to be able to do is whenever you see a sugar, to be able to identify basically your either hemiacetal or hemiketal. Because if you remember, an aldehyde, oh, gosh dang it, aldehyde plus alcohol equals hemiacetal. A ketone plus an alcohol equals a hemiketal. Now, I'll go back a couple of slides because did we have that? Did we look at a aldehyde react with an alcohol? Yes, we did. This aldehyde right here reacted with this alcohol, and that cyclized our sugar. So one of the things that's kind of tricky is, well, which is the species or which are the atoms that reacted? Well, this carbon has a partial positive charge. This alcohol has a partial negative charge. That oxygen of that alcohol is this oxygen right here. The carbonyl oxygen becomes our hydroxide group. Okay, so whenever you're looking to identify a free reducing end, I recommend looking at it as basically OCO, oxygen, and then a carbon, and then an OH. So our oxygen within our ring, followed by a carbon, followed by a hydroxyl group. And if you're kind of confused as to how to do that, well, find that oxygen and say, okay, well, do I have an OH group next to that carbon? Okay, here's my oxygen, here's my carbon, here's my OH group. Good, I got it. Now, looking right here, here's my oxygen. Sorry, looking in the middle, I've got my oxygen, here's my carbon. Then, is that an OH group? No, it's not. That's an oxygen that has two single bonds. So that oxygen is connecting one sugar to the next. When we go on to this, oxygen, carbon, OH group. Okay, that's a reducing end. So then if we look at this, oxygen, carbon, oxygen connecting each, oh, not an OH group, so not a reducing end. So with that said, we have this terminology related to sugars. If we see a free reducing end, we would classify that sugar as a reducing sugar. By virtue of it having one reducing end, that makes the entire sugar a reducing sugar. If it has no reducing ends, then it is a non-reducing sugar. Now, thinking about that and looking at ribose, okay, ribose, that's a monosaccharide. Your cyclic monosaccharides, like what we see with glucose. Is glucose a reducing or a non-reducing sugar? Add it to the chat. R for reducing and N for non-reducing. Looking at that cyclic, that's right. It's a reducing sugar because it has that OCOH. Okay, great job, everyone. Now, we can look at a sugar and be able to identify it as a reducing or a non-reducing end, or a reducing or a non-reducing sugar. And we look at disaccharides. There are three disaccharides that I want you guys to know. I want you to know sucrose, lactose, and maltose. Those are the three disaccharides that I want you to know. Are there other disaccharides? You betcha. There are plenty of other disaccharides, but these are the three that I want you to know. Now, when we look at this right here, this is sucrose. Sucrose is a disaccharide made up of glucose on the left and fructose over there on the right. Now, we've got our sugars numbered here. We've got all of our carbons numbered O, C, O. Okay, well, at very least, glucose does not provide a reducing end. Then if we go over to fructose, oxygen, carbon, oh man, that oxygen, not a reducing end. So what that means is, or one thing that's worth recognizing about this is that how did we classify fructose? As an aldose or a ketose? Add A to the chat for an aldose or K to the chat for ketose. We added, it was a ketose. It is a ketose. Now, what that means is when you think about a ketose, which carbon is your carbonyl carbon? Thinking about it in its linear form, it's carbon number 2. So our partial positive charge is right here. So this carbon links up with this oxygen. So our ring is a little bit smaller. It's a 5-membered ring. When fructose cyclizes, it doesn't form that 6-membered ring. It can, but chances are it's going to form a 5-membered ring. It's going to form a ketofuranose because it's a ketose and it's a 5-membered ring. Now, what this also means is thinking about that carbonyl carbon number 2. This is carbon number 2 now. When we looked at the linear form of of fructose, was carbon number 2, where that functional group, that carbonyl carbon, was that a chiral carbon? It was not. It was not a chiral carbon. But now, all of a sudden, the carbon that I have circled there, let me ask you this and answer Y for yes and N for no. Is the carbon that I have circled there, is that a chiral carbon? Yeah, it is. It's bound to 4 different things. So it's a chiral carbon. Not only is it a chiral carbon, arguably it's a new chiral carbon. And what's the term that we use to identify a new chiral carbon? It's an anomeric carbon. So what this means is that glucose and fructose, these are forming a linkage where it is an anomeric carbon to an anomeric carbon. Now, the significance of that is that, well, how did we classify this? Was this a reducing or a non-reducing sugar? It was a non-reducing sugar. When we have the anomeric carbons form a linkage with one another, we make a non-reducing sugar. Now, this specific compound, oh, dang it, Joel, what did you write there? This is an alpha 1 beta 2 glycosidic linkage. Now, this is an instance where the alpha and beta configuration is a little bit challenging and a little bit tricky. When it comes to drawing a sugar, if you have the glucose with this hydroxide group pointing downward, I mentioned the other day that that was an alpha carbon, or that's the sugar in its alpha configuration, and I stand by that. One of your classmates asked, well, how does that relate to carbon number 6? Well, provided that the hydroxide group is pointing down and carbon number 6 is pointing up, then that is absolutely true. When it's pointing down, it's alpha. Now, if we look at fructose here, what do you notice about the direction? And just for a moment, let's forget about this. There's a hydroxide group. Would everyone agree with me that that hydroxide group, add a Y or an N to the chat if you agree with me, an N if you don't agree? This hydroxide group, is it pointing down from our ring? Great. Yes, it is. Okay. Now, using that same logic, how would you describe carbon number 6? Is that pointing D for down or U for up? Your eyes don't deceive you. It's pointing down. Okay. So this is an instance where we kind of have to go to that next level definition. Our hydroxide group on our anomeric carbon is pointing in the same direction as carbon number 6. Since they are pointing in the same direction, that is considered beta. If they are pointing in opposite directions, then that is considered alpha. Now, what I said or one of the things that I mentioned the other day was, well, you can use that explanation of down is alpha because I would say 90 times out of 100, carbon number 6 on a structure of glucose is going to be pointing up. And so then you can look at it and be like, oh, well, my hydroxide group is pointing down, therefore alpha. Down is alpha. I'm good. You do have to reference back to that carbon number 6, but it's somewhat rare. So this is an instance where we are applying that, though, in this specific structure. Now, what I want you to take away from all of this is I want you to know the following things. One, what are the components of sucrose? Well, it's glucose and fructose. Next, I want you to know, I'm going to erase that and put it over here. The second thing that I want you to know is that, well, it is a glycosidic linkage. That connects these two sugars to one another. And third, I don't want you to stress too much about this being an alpha beta linkage, but I do want you to know that alpha and beta notation, I'll say, is used when describing linkages. So those are three things that I do want you to know about this. Now, another way that you might see sucrose would look a little bit like this, where we have this alpha glucose basically stacked right on top of that beta sucrose. Again, our hydroxide group on carbon number 2, our anomeric carbon of fructose, it's pointing up, but so too is carbon number 6 pointing up. So they're both going in the same direction, which is why we give it the beta notation. All right. Next up is lactose. And again, kind of thinking about, well, how would we identify this? Okay. What I would like you guys to do is look at this, and I got a couple of questions, and I just want to see responses in the chat. I'm going to say type of sugar. R for reducing or N for non-reducing. Take a minute and look at that whole sugar. See lots of R's. Great. Okay. I would agree with that statement. This is a reducing sugar. Now, does it have a non-reducing sugar? Absolutely. We've got OCO, not OH. So that's a non-reducing end. Okay, but then over here we've got OCO. Okay, so that would be the where the non-reducing end is. Now, my next question is this an alpha or beta reducing end? And then A or B. Great job, everyone. It's beta because we have this pointing up, so too is carbon number 6 pointing up. Okay. Now, when you look at this sugar, I think that it's really easy to kind of get distracted by something. For instance, we've got carbon number 2, 3, 4. Okay, then we got carbon number 2, 3, and 4. Um, Respond in the chat, 1 or 2. Which one of the sugars, 1 or 2, is glucose? Okay. I'm seeing a really good mix of people saying 1, and lots of people saying 2. Okay, it's 2. Okay, so we need carbon number 2 to be down. Carbon 3 is up. Carbon 4 is down. Over here, this other sugar that I've labeled 1, this is carbon 2, down. Carbon 3 is up, and carbon 4 is up. Now, with that said, do we know what is the relationship between glucose and galactose? I mean, they're friends, but what's the term that we use to describe them? They are epimers. They are C4 epimers of one another. Now, one of the things that I've noticed over the years of teaching this class is that sometimes people will look at a structure and they'll say, oh, well, you know, this right here, this sugar that I've drawn a big circle around, they'll, sorry, let me rephrase this. What they'll do is they'll tend to look at the linkage, the glycosidic linkage, and they'll think that this bond pointing downward is insignificant. And it is absolutely significant. This pointing down, like what I would recommend if you're ever looking at a sugar and you're like, which one is that? What I would do is scribble out the other sugar and throw an H on there and say, is this glucose? Well, down, up, down. Yes, that's glucose. Because the reference point or the way that you draw that linkage and the way that you draw those individual sugars is important. So this is galactose and this is glucose. There we go. That makes up lactose. What type of linkage is this? Well, this is a beta. Now I'm going to use numbers to indicate the linkage. How would you describe what carbons are involved in that linkage? 1,4, a beta 1,4 linkage. Great job. Great job. In the words of Stone Cold Steve Austin, hell yeah. Okay, we've got a beta 1,4 glycosidic linkage that connects those two sugars from one another. I'm sorry for swearing, but Stone Cold Steve Austin and disaccharides just get me all pumped up. So we've got a beta 1,4 glycosidic linkage that connect these two sugars to one another. Glucose on the right, galactose on the left. I think that lots of people saw the parentheses and saw glucose plus galactose and looked at that and said, aha, that's what I'm looking at. I'm reading left to right. I'm going to read this structure left to right. I didn't do that as like, haha, this is a teaching tool. I'm going to teach the heck out of them. Restart for number of the carbons. I can absolutely go over where I number the carbons. Okay, so okay, I'm going to draw cyclic galactose. This is going to be galactose. So what I need is carbon number 2, hydroxide on the right. 3 is on the left. 4 is on the left. And then 5 is on the right-hand side to make it a D sugar. Okay, so there is my general structure for galactose. Now, carbon number 1 right here is my aldehyde functional group. And whenever I cyclize this, this has a partial positive charge. This has a partial negative charge. So the lone pairs of electrons on my oxygen up here are going to do a backside attack. The mechanism is going to push these electrons up here. Oh man, I'm all out of organic chemistry terminology. But that's what's taking place. We got those lone pairs of electrons on that oxygen that are going to act as my nucleophile, attacking my carbon. That's my electrophile. My structure is going to look a little bit like that's not a very straight line. But now I've got this. Boom, 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 boom. There's my 6-membered ring. Oxygen is one of those members. The oxygen that is right here is this oxygen that had the lone pairs of electrons. Carbon number 6 is right next to that or very close to that, and it's going to be right here, CH2OH. Okay, now carbon number 1 is right here. That carbon number 1, which was where my aldehyde functional group was with my carbonyl, that now has picked up a hydrogen, in which case OH. Throw down right there. I'm pointing downward on this, in which case I'm looking at an alpha sugar because that is in the opposite direction as my carbon number 6 is relative to my ring. Now, like I said, I'm drawing galactose. So carbon number 2 right here that I just drew a circle around is this carbon right here, number 2, that now my OH is pointing downward. Carbon 3 is up because on my linear form it's on the left. And then carbon 4, my hydroxide group on the left, is going up right here. Okay, so that's my quick review on lactose. Why doesn't this OH on C2 have a lone electrons that are... I got a private message that says, why doesn't the OH on C2 have a lone pair of electrons that are attracted to the functional group? Well, I'll tell you, it does. But if you think about that for a moment and kind of, sorry, I wasn't intending to sound like, well, if you think about that, I hope that it didn't come across that way. And now that I bring it up, now I feel like I didn't sound like that, but now I'm second guessing myself. Okay, so you bring up a very good point. Because every single one of these hydroxide groups has those lone pairs of electrons. If this lone pair of electrons goes right here and attacks that carbonyl carbon, yeah, we have those partial charges, in which case we could have that. But think about this is the point where I'm like, I'm stressing to, imagine what you're making. You would be making a ring that's basically an epoxide. It's a 3-membered ring, not a particularly stable ring. If these electrons right here attacked there, could that happen? Absolutely. How large would that ring be? Well, that would be a 4-membered ring. Your ring would consist of 1, 2, 3, and the oxygen would be your 4th member. Then an alternative is if this hydroxide group on carbon number, that would be carbon number 4, could that attack? Yeah, it could. Would you form a ring? Yeah, you would. How large would that ring be? 5 members. Well, 5 is more stable than 4. 4 is more stable than 3. So the person who sent me this message asking about could these hydroxide groups attack, they absolutely could. But it has to do with the stability of the ring that results. And a five-membered or a six-membered ring is typically going to be stronger, which is why they're going to kind of emerge as the most likely outcome. Um, so the most likely, the most sterically favorable ring is going to be that six-membered ring or a five-membered ring. Thank you for the question. That's a fantastic question. Okay, so I've addressed those two things. I presume that those are good to go. Now I'm going to go on to look at lactose. And if I didn't answer your questions enough or adequately, just let me know and I will get back to them. So here is lactose, or here is maltose. Maltose is a disaccharide of glucose and glucose. It's a fun one. Now, looking at that one, is this a reducing or a non-reducing sugar? Add an R if it's a reducing sugar or an N if it's a non-reducing sugar. You're right. It's a reducing sugar because we have this free end here. Now, my next question is this is a glycosidic linkage. We've got carbon number 1 and carbon number 4. So this is a 1,4 glycosidic linkage. How would you classify this linkage, alpha or beta? A for alpha, B for beta. Good job. Great job, everybody. You're doing a fantastic job. So this is an alpha 1,4 glycosidic linkage that connects these two. Now, one question that I always like to ask is, okay, sure, this is an alpha 1,4 glycosidic linkage. Does that have any influence over the configuration of my reducing end? Here I've got both of those carbons are displayed in the alpha configuration. Is there anything stopping this ring from opening up and then this hydroxide group swinging up and then the ring closing again? And this converting from the alpha to the beta configuration, is there anything stopping that? No, there's not. So you can have sugars that are, yeah, there's an an alpha glycosidic linkage that holds them together, but this would be the beta form. Your reducing end. Your reducing end is going to be the end that is dictating, well, what form of sugar is there. So when you're thinking about this in terms of exam questions that I'll ask you, my exam questions, or I'd like to think that the exam questions that I will give you guys are going to be targeted, and I'll say something like this linkage, and I'll give you a couple of different options. One, or A, B, C, D. I'll say alpha 1, 4, alpha 1, 1, beta 1, 4, and beta 1, 1. What type of linkage, or what is the best description of that linkage that's there? And then your answer would be, okay, well, it's an alpha linkage, so these two are out. And then it's carbon number 1 going to carbon number 4. Okay. So this is maltose, glucose and glucose, alpha 1,4 glycosidic linkage that connects the two of them. Now, we're getting into some polysaccharides. Why? Because they're fantastic. Cellulose, glycogen, and starch. Um, Cellulose, something you've probably heard of. It is a structural polymer for plants. It's found in the cell walls of plants as well as in some bacteria. Now, what I want you to notice about this is, well, let's just look at this right here. What sugar is that? And imagine the box expands just a little bit. What monosaccharide is that in that box? We've got a pattern of carbon 1, 2, 3, 4. We got down, up, down. And, you know, I got to be honest with you. The, again, excuse my language, but the kick-ass Metallica bond that we've got going there is always a little bit distracting because this is one instance where organic chemists might be like, you can't do a bond like that. This is not a carbon, so don't be distracted by that. Instead, what this funky little bond is trying to show is basically that this is pointing down, but this sugar is in the beta configuration. So this is all kind of like an artist rendering sort of situation that presents the problem. Now, with that said, does anyone want to say what sugar that is right there? Glucose. It's absolutely glucose. So glucose, this is a polymer, or it's a polysaccharide of a bunch of glucose molecules, a bunch of glucose monomers. Now, what type of linkage, or how would you describe this linkage? It's a glycosidic linkage. How would you describe it? Beta 1,4. It's a beta 1,4 glycosidic linkage. That right there, that rotation, that beta configuration is why I can't go outside and find a tree, pick some bark off, just start eating and getting some calories from that. Because that beta configuration is why is something that prevents the enzymes that are in my body that are responsible for breaking sugars down, polysaccharides down, can't do anything with this. These beta 1,4 glycosidic linkages, one, the enzymes that are in my body can't do anything with that, but also that beta configuration makes these like sheets of cellulose have this really intricate and strong um, hydrogen bonding network, which makes cellulose even more strong. So you've got beta 1,6 or beta 1,4 glycosidic linkages. So glucose after glucose after glucose. So cellulose has tons of energy or tons of glucose, which you could convert to energy, but these beta linkages kind of prevent, us from using it in any sort of form or fashion. I mean, you can burn it and stuff, but I mean, like dietarily or nutritionally. Glycogen. This is a short-term energy storage in animals. It's found in the liver and in the muscles. Now, this molecule, it's just a bunch of glucoses over and over and over again. The difference between this and or the significant point of glycogen is that it has both alpha 1,4 and alpha 1,6 glycosidic linkages. These alpha 1,6 glycosidic linkages are sometimes referred to as branch points, and there are multiple branch points with multiple branch points found throughout a glycogen molecule. And the cool thing about glycogen, and I'm going to switch my screen share for a moment because I always like showing this picture. What does a glycogen molecule ultimately look like? This right here is a depiction of glycogen. At the center is a protein known as glycogenin, but then all of these, this kind of network of, I don't know, I guess it almost looks like a spider web of sorts. Those are glucose chains that go on to extend to make a glycogen molecule. So we've got tons and tons of branch points. And then we have basically, well, a glycogen molecule has one main chain, and then it just has branch points off of branch points off of branch points off of branch points. Now, with that in mind, imagine that this right here is an OH group. So I've just drawn a circle there. How would you describe that end? Probably as a reducing or a non-reducing end. Would you say it's an N for non-reducing or an R for reducing end? It's a reducing end. Perfect. Okay. Now, over here, R or an N? N. Here, R or an N. Yep. Every time that there's a branch point, every end of a branch point is a non-reducing end. So one of the things that's significant and interesting about a glycogen molecule is it has, or a glycogen molecule has one reducing end, and then number of BP or branch points, or actually number of branches plus one equals the number, or sorry, yeah, number of non-reducing ends. Okay, now that's what I wanted to get to. Glycogen, glucose after glucose, it's all stored up there. Alpha 1,4 and alpha 1,6 linkages are the main important parts about that. Why is this? I don't know why I did that. Okay, now starch, which consists of amylose, which is a linear polysaccharide. It is a linear molecule that's just a bunch of glucoses over and over and over and over again. A starch molecule is typically 300 to 600 glucose subunits long. And this is a short or a long-term energy storage molecule in plants. It's got a bunch of alpha 1,4 glycosidic linkages. It's glucose after glucose. But whenever you talk about starch, you're really kind of talking about two different sugars that are kind of intermingled with one another. One of them is amylose, and the other one is amylopectin. Amylopectin and amylose are kind of threaded together with one another, which makes it a little bit more difficult for your stomach to digest them. If you look at the structure of amylopectin, what types of linkages do you see? Yeah, it's repeated glucose molecules, but well, they're alpha 1,4 linkages and alpha 1,6 linkages. So the linkages between, or the linkages that exist within amylopectin are the same as what exists within glycogen. One of the differences, however, though, is look at this down here. This is indicating that between branches, so we've got B1, I'll say, and then B2, branch number one and branch number two. There's between 30 to 50 individual glucose subunits that basically separate those from one another. Now let's go back and look at glycogen. Our glycogen branches are separated by anywhere from 7 to 11 glucose subunits. So the branches in glycogen are much closer together than the branches in amylopectin. Now, the difference and the significance of that is, well, that's because that gives amylose the ability to kind of thread itself through these branch points. And those two molecules are going to interact with one another. Okay, it's alpha 1,4 and alpha 1,6 glycosidic linkages, just like glycogen. There we go. Now, one of our next polysaccharides is chitin. Chitin is a molecule that is beta 1,4 glycosidic linkage, or beta 1,4 glycosidic links, glycosides of N-acetylglucosamine. And so that would be, we have carbon number two. So remember, two is down, three is up, four is down. And then we've got our modification to that. This is a strong polysaccharide found in the exoskeletons of insects. And you could think about this almost like cellulose, but with an N-acetylglucosamine instead of glucose. So N-acetylglucosamine is the repeating unit over and over again. Now, there we go. So now what we're going to look at are, okay, actually, before I proceed, Does anyone have any questions about polysaccharides? The important polysaccharides would be glycogen, um, amylose, amylopectin, and cellulose, and chitin, those five. And this is, I guess, if you're, let me do kind of a little bit of a, takeaway from all of this. I do want you to know the similarities and differences between these different polysaccharides. For instance, amylopectin is very similar to glycogen. What makes them different from one another is the frequency of the branches. Glycogen has a lot more branches than amylopectin. So, one of the things that I always like to kind of ponder is, well, if your body used amylopectin as a glucose reserve as opposed to um, glycogen, well, your glycogen store would have to be considerably larger to have the same amount of energy. So, in comparison between amylopectin, or a comparison between amylopectin and glycogen, those are two sugars that, well, they are calorically going to be considerably different. The frequency of the branches in glycogen give us the ability to run a marathon. If somebody had amylopectin instead, they're not going to be able to run the whole marathon, or they're just going to have to, carb load. Now, amylose. What is amylose most similar to? Well, amylose is linear. What's another sugar that's linear? Getting there, getting there. Cellulose. Cellulose is a linear sugar. The difference between cellulose, however, and amylose is the linkages. Cellulose has beta linkages as opposed to the alpha linkages. Okay. So I want you to know the similarities and differences. I want you to, you know, if I showed you a structure, I'd want you to be able to say, oh, well, this is a polymer of this specific sugar. This is this type of linkage that connects those two. Now, I also want to point out sometimes whenever it comes to linkages in some text, you might see a beta linkage like this drawn in this fashion, CH2OH. And this is one I think that organic chemists have a real strong position and a real strong justifiable opinion to say, well, that's just absurd. You'll see them drawn like this. And that's an instance where I'm like, I think we might be fudging the rules a little bit too much as biochemists. But what this is really trying to display is that this hydroxide group on carbon number 4 is pointing down, and this hydroxide group on carbon number 1 is pointing up. It's in the beta configuration. Okay. Now, next up, glycosylation. What are glycoproteins? Glycoproteins are proteins that are linked to oligosaccharides. There are two major types of glycoproteins. They are N-linked or O-linked. Okay. Now, what this means is that if you think about the central dogma of molecular biology or the central dogma of biochemistry, there's DNA that goes to RNA that goes to protein. Okay. Now, that's a real nice, simple story. The process by which RNA is converted to a protein is known as translation. Okay. We make a protein that has, 400 amino acids, and okay, it's got all these different amino acid R groups that they can interact with this and this and this. There's a process known as a post-translational modification. So those amino acid R groups can be modified after translation. Now, those amino acid R groups, when they get modified, that changes their makeup. One of those post-translational modifications is glycosylation, or the addition of a sugar to a protein. So imagine it is a protein has been translated, has been translated. And when that protein has been translated, then there's an enzyme that comes along and it says, hey, I see an asparagine there. That's neat and well and everything, but I feel like we should throw some sugars on there. Or there's a protein that has either a serine or a threonine and says, those are neat. Those are nice polar uncharged residues, but I think it'd be much more interesting if we threw a bunch of sugars on there. And these glycosylations are a really fascinating aspect of biology and developmental biology. If you've ever heard of Niemann-Pick disease or there's not beriberi, Guillain-Barré, I think. There's a number of different um, diseases that are genetically linked that people have that are related to glycosylations and the lack thereof, or the kind of um, distorted processing of a sugar from a protein. Now, with that in mind, we also are going to look at glycosaminoglycans and peptidoglycans. But for right now, we're looking at glycosylations. Glycoproteins are proteins that have these oligosaccharide links. Okay, so imagine that we have a protein with a particular amino acid, let's say a serine or a threonine, and basically a long, elaborate network of sugars are going to be added to it. And N-linked glycosylations, those are going to happen in the ER, also known as the endoplasmic reticulum, or the Golgi apparatus. So lots of post-translational modifications are going to take place in either the ER or the Golgi. So after translation has occurred, these are kind of like the finishing spaces. Like, yeah, the car's been made, but let's paint it. Let's put some flames on it. And what I want you to take away from this is that this, or sorry, all of these are different sugars that can be added to a protein. We've got mannose, glecnac, which is our N-acetylglucosamine. Um, I can't remember what new 5GC is. Galactose, sialic acid, xylose, and fucose. These are all sugars that can be added. And the significance of this, the importance of this is that these different kind of patterns or the way that these sugars are added and what they look like, they're almost like a messenger of sorts. Um, they're, like I said, these are very important steps in developmental biology, and they're related to some heritable diseases. Okay, so what I want you to know about glycosylation is the amino acids that are likely the targets and where these glycosylations take place. N-linked glycosylations, asparagine. Okay, now this should take you back to looking at the structure of asparagine. What do we have on asparagine? Well, asparagine looks like this, CH2. This is just the R group. That's what our asparagine R group looks like. We've got that NH2, hence N-linked. So this is going to be where a sugar is going to be added. And so we'll have something like CH2OH. And then from there, we'll have another sugar that's added. And then from there, another sugar that's added. And depending on the pattern there, that's going to be important for like a developmental checkpoint. Okay, next up, and I believe this is going to be the last part of our class period today, describe the roles of glycosaminoglycan polysaccharides in organisms and cellular processes. Now, we've got, well, a glycosaminoglycan is what is known as a polyanionic polymer of a modified sugar. So this one right here, hyaluronic acid. Well, if I were to ask you a question about this, I would show you this and I could ask you, well, what sugar is this that gets modified? Again, carbon number one, down, up, down. That's glucose. What has happened to it though? Well, we replaced that CH2OH with a carboxylic acid. I could also ask you, what configuration is this in? That's in the beta configuration because we've got it pointing up. So this is hyaluronic acid. Another glycosaminoglycan is chondroitin, and then another one is heparan sulfate. These are all three different glycosaminoglycans. Now, what's the significance of them? Well, one thing that they are involved in is they are involved in inflammation or inflammatory responses. Heparan sulfate is one glycosaminoglycan that's involved in blood clotting and activating blood clotting. Another example of a glycosaminoglycan is that they make the extracellular matrix. So outside of a cell, the matrix that exists, they help to make it a more gel-like structure. So what they do is they hydrate the extracellular matrix. So if you're thinking, okay, what the heck am I supposed to say about glycosaminoglycans? One thing that you can say is they are polyanionic polymers of modified sugars. Take the party line, say the definition. Another thing that you could say is, well, they're involved in blood clotting. And then another thing that you could say is you could say, well, they help to hydrate the extracellular matrix. So those are important parts about glycosaminoglycans. Next up, peptidoglycans. I really love saying that word because I remember whenever I was a freshman, my biology professor said peptidoglycans. And I was like, I don't feel like that's how you say it. So I'm going to swing the pendulum in the opposite direction and try to pronounce every single letter in that name. So peptidoglycans. Here we are. Peptidoglycans are peptides or protein linked to polyanionic polymers of modified sugars. If you've taken a microbiology class or lab, you do that gram staining and you stain it and you say, aha, it's purple. And then you look around the room and you're like, why is only mine purple? Yeah, everyone else must be wrong. Yeah. Um, Point is, peptidoglycans, well, they help maintain that extracellular matrix, and it gives you that idea of, classification for different types of bacteria or bacterium. They are complex structures. They provide strength and shape to cell walls of bacteria. Okay. One of the things that is kind of a pattern or an important part about peptidoglycans is you've got that repeating network of NAG and NAM, and that is N-acetylglucosamine followed by N-acetylmuramic acid, NAG, NAM, and it's just those sugars repeated over and over and over again. Now, I want you to remember NAG and NAM, and not necessarily I've got the structure committed to memory, but instead, later on this semester, we'll be talking about enzyme mechanisms. And we'll bring back NAG and NAM because we'll be talking about an enzyme that's responsible or capable of grinding those up or breaking those up. So, with that in mind, today and Tuesday and Monday, last Monday, the other day, the day before yesterday, we've covered a lot of stuff. I want you to be patient with yourself. I want you to remember that I believe in you. I want you to also think about, okay, what are the important things that I should know about this specific subject? Should I know about the structure of every single aldose? No. Should I know the structure of every single aldohexose? No. If he shows me an aldohexose, should I be able to identify it as glucose or galactose? Yes. If it's not glucose or galactose, could I relate it to glucose or galactose? Is it a C2 epimer? Is it a C3 epimer? Those are good things that you should be able to do. I plan on having your practice exam up for this next exam before the end of the weekend so that you'll have something of like, okay, these are good questions that I should be able to look at. I've got your quizzes up for this week. If you have any questions about those, please reach out to me. When is the next exam scheduled for? The next exam is on, one person said it's not until April. And I think that's about right. Yep, at the very beginning of April, because we've got one at the beginning of April, another at the end of April, and then we've got our final exam. Um, Now, with that in mind, um, we've got a lot of information, and yeah, I think that yeah, it's a recording from has you could have sent that question as a private message, but instead you embarrassed me on all the things that I haven't done. Oh my gosh. It's okay. No, it has not. I will post it. I'm gonna, a friend of mine who's an attorney, he always says, you you promise low and then deliver high. So I will have Monday's lecture up by July. That's my promise. July. So that means any day before July, I'm good. Just checking so we have B1. Yes. Sunday you've got quizzes B1 through B4. Remember, B1 is an extra credit one, but I'm going to do that extra credit on the back end. Then activities 9 and 10. Um, yeah, I think cool. Like I said, next week, asynchronous. If you got any questions, reach out to me and I'll be able to meet up with you for an office hour or something. I'm glad to do that. And then, if you get bored over spring break, you're like, hey, I was thinking about biochemistry. What's up? I will post Monday's lecture before July. That was my snarky comment. Yes, I will post it. But if you got questions over spring break, I can do office hours then as well. Just let me know. What? Throwing an ASAP at me? Okay, everybody. Thank you very much for coming to class. I'm happy that I recorded that last little bit where I shared my secrets to promise low, deliver high.